All right, let's talk about a technique that really changed the game in molecular biology, real-time PCR. It takes the incredible power of DNA amplification and turns it into a tool that's not just powerful, but also amazingly precise and quantitative. So let's break down how it all works.
So, here's our game plan. We're going to kick things off with the basics, the foundation of conventional PCR. Then we'll see how real-time PCR is a huge step up. After that, we'll dive into the clever chemistries that let us see everything happening in real time, unpack all the key advantages, and wrap up by looking at how critical this is in modern diagnostics.
Okay, so before we can really jump into real-time PCR, you've got to understand where it came from. And that's the original polymerase chain reaction, or PCR, dreamed up by Kary Mullis back in 1983. This is the engine that drives everything else we're going to talk about.
At its very heart, PCR is just a simple three-step cycle that repeats over and over and over again. First up, denaturation. We crank up the heat to about 95 degrees Celsius, and that heat literally unzips the double-stranded DNA. Next, we cool things down in a step called annealing. This lets these little DNA sequences called primers find and bind to our target. And finally, extension. A tough, heat-stable enzyme called Taq polymerase gets to work building brand new DNA strands. You do this 30 or 40 times, and you get this incredible exponential amplification.
And this is where the magic really happens. Take a look. In just one cycle, one copy of DNA becomes 2. In the next cycle, those 2 become 4. It just keeps doubling. It's an exponential explosion that can turn a single molecule into 10 million copies or more. That gives us plenty of material to actually see and detect.
But, traditional PCR had a big drawback. You could only see the final result at the very end of the whole process. And that's exactly where real-time PCR comes in. It's a massive upgrade because it lets us watch the amplification as it happens, cycle by cycle, in real time.
So what's the big deal? Well, here's the fundamental difference. Traditional PCR, it's like taking a photo at the end of a race. You only measure the final product after all the cycles are done, in what's called the plateau phase. But real-time PCR, it's like having a video of the whole race. It collects data during the exponential phase, which is when the amount of DNA is perfectly doubling each cycle. This is a total game changer for accuracy.
This graph right here, this is probably the most important concept to grasp. That green line climbing steeply, that's the exponential phase. It's pure, it's predictable, and it's exactly where real-time PCR gets its data. Traditional methods, they wait until the plateau at the top, where the reaction is running out of gas and things get messy. That makes the results way less reliable if you're trying to figure out how much you started with.
So that begs the question, right? How in the world are we seeing this DNA being made in real time? Well, the answer is a little bit of molecular light, fluorescence. And we get that glow using some really clever chemical methods, or formats.
Let's start with the simplest approach, a dye called cyber green. Now this dye has a cool trick. It only lights up, it only fluoresces brightly when it's bound to double-stranded DNA. So it's simple. As each PCR cycle creates more double-stranded DNA, more dye latches on, and the signal gets stronger and stronger. It's a direct, easy measurement. The main benefit? It's simple and cheap. The catch? It'll bind to any double-stranded DNA, not just the specific piece you're looking for.
And if we zoom way into the molecular level, you can see exactly what's happening. That little green molecule, SYBR green, is literally slotting itself, or intercalating, right into the DNA's double helix. The more DNA you make, the more slots there are, and the brighter the whole tube glows.
Okay, so what if you need to be absolutely sure you're only detecting your specific target? That's where things get more sophisticated, with tech like FRET. That stands for Fluorescence Resonance Energy Transfer. Instead of one simple dye, this uses 2 different, highly specific probes that are designed to bind right next to each other on your target DNA, and only on your target DNA. So check this out. We shine a light to get a donor dye excited, but instead of just fluorescing itself, it passes that energy over to a nearby acceptor dye, which then lights up. Here's the key. That energy transfer can only happen if those 2 probes are bound side by side, which ensures we're not just detecting any old DNA, but our specific sequence of interest.
All right, now this next one, TaqMan probes. This is seriously clever. It's also known as a 5' nuclease assay, which sounds complicated, but the idea is brilliant. It basically hijacks the natural chewing action of the Taq polymerase enzyme and uses it to create our signal. It's just a fantastic piece of bioengineering. So here's the setup. You've got this special probe with 2 dyes on it,
a reporter, which is the one that can fluoresce, and a quencher, which acts like a light switch, keeping the reporter turned off as long as they're close together. When the probe is just sitting there bound to the DNA, the quencher is doing its job. No signal, darkness.
But here comes the cool part. As the Tag polymerase moves along, building the new DNA strand, it literally runs right into the probe. And using its natural ability, it just chews that probe to pieces, physically cutting the reporter dye loose from the quencher. Once it's free, the reporter can finally light up, giving us a flash of light that's directly tied to a new copy of DNA being made. Just elegant.
And finally, we get to what is probably one of the most sophisticated designs out there, scorpion primers. These things are remarkable. They actually build the primer that starts the reaction and the probe that detects it into one single, beautiful molecule. This design makes the whole detection process incredibly fast and super efficient.
So how does this little marvel work? Well, first, the scorpion primer binds to the DNA and gets extended by the polymerase, just like a normal primer. Now it's actually part of the new DNA strand. Then, in the next cycle, as things cool down, the probe part of that same molecule folds back on itself and binds to a sequence on the very strand it just helped create. It's an intramolecular reaction, meaning it binds to itself, which makes it lightning fast.
And this image really brings it all together. On the left, you see the scorpion in its off state. It's folded into a tight little hairpin loop, which keeps the reporter and quencher dyes snuggled up close. No signal. But on the right, after it's bound to the newly made DNA, that hairpin is forced to open up, separating the reporter from the quencher, and boom, you get that beautiful fluorescent signal.
Okay, so we've just waded through some pretty clever chemistry. But let's take a step back and ask the big question. Why? Why go through all this trouble? What really makes this real-time approach such a massive, revolutionary leap forward?
Well, the advantages are just huge. First, you're getting super precise data from that clean exponential phase, not the messy plateau. The results are fast, often under 2 hours, and you can completely skip all the slow, tedious post-PCR work like running gels. Plus, because the whole reaction happens inside a sealed tube, the risk of contamination just plummets. And all of this together makes it an incredibly powerful tool for easy and accurate quantitation.
So with all these incredible benefits, it's really no surprise that real-time PCR has become an absolute cornerstone in clinical diagnostics, where speed, accuracy, and sensitivity are everything.
Think about virology, for instance. It's a game changer for quickly spotting and counting DNA viruses. It lets doctors rapidly identify common bugs like herpes simplex virus and adenovirus. And it lets them monitor the amount of virus, the viral load, for diseases like hepatitis B, so they can see if a treatment is actually working.
And it's not just for DNA viruses. It's just as powerful for RNA viruses too. You just have to add one little step at the beginning called reverse transcription, which turns the virus's RNA into DNA. This RT-PCR method has been absolutely critical for public health, giving a sensitive test for things like the SARS coronavirus, enteroviruses, and hepatitis C. So that really brings us to the future, and a final thought to leave you with. We're already seeing the rise of multiplex real-time PCR, the ability to look for many different targets in a single tube at the same time. So the big question is, how is this going to completely transform our ability to find and quantify multiple pathogens in a single rapid test? The potential is just immense, and it's definitely a space worth watching. Thanks for joining.